our first session for today is the Millennium Science Roots section. So this is a particularly special session for us because at the core of what we are as a company is that we're all scientists. So everyone from top to bottom in this company has some type of science experience, whether it's a higher educational degree, a postgrad degree, doctoral degree, or they have spent significant time in the lab. And as such, we have maintained connections with the lab groups that we have come from, because we're genuinely interested in the science and the technologies that come out of some of these labs. And so it's a very special honor for me to introduce our first speaker today, Dr. Lola Mugisho. Lola and I did our PhDs together at the University of Auckland in the Department of Ophthalmology as part of the Buchanan Ocular Therapeutics unit. And since completing her PhD, Lola's gone on to publish numerous articles, presented many conferences, and is now the deputy director of the lab group that we started in Bochu. So she's had a fantastic career and she's only just starting. So without any further ado, take it away, Lola. The stage is yours.
Well, thanks for the intro, Fraser. It's really good to be here because it's been a while since we've presented at any conferences. So this is a unique and treasured opportunity. So I'm just going to share my screen, share my talk. Just give me a moment. That's done. Okay, can everyone see my screen? Oh, sorry, let me change that display setting. Yeah, can everyone see my talk now? All right. Yeah, cool.
Yeah, perfect.
So today I'm going to talk about some of the work that we've done. Obviously, a 15-minute talk doesn't cover everything. So if you have any questions, please feel free to email me afterwards. But the title of my talk is The Inflammasome Pathway in Chronic Retinal Diseases. So the inflammasome is a part of what's known as the innate immune system. This is the first line of defense, and it's usually non-specific, so responds to pretty much any danger or pathogen that the cell experiences. And this pathway has been implicated in several chronic diseases. So the graphs I've shown here on your right is just to give you an idea of how the inflammasome research world has really grown over the last over 10 years, from 2010 to 2020. So what you'll see is that in 2010, the number of articles published on inflammasome were about maybe 3,000. By 2020, we were looking about 16,000 per year of published articles on the inflammasome. So obviously there's been a huge growth in the area. But when we look at the eye specifically, the growth has actually been more exponential, going from less than 200 in 2010 to about 2,600 in 2020. So a lot of work has gone in over the last decade or so into inflammasome research. And this has actually been mostly targeting this pathway for treatment of chronic diseases. For us, the type of inflammasome we're particularly interested in is the NLRP3 inflammasome. And this is because it's the most studied and it's the pretty much most implicated in diseases as well. The NLRP3 inflammasome usually requires a two-step activation process. The first process is the priming step, and during the priming step, you have the three main components of the inflammasome, NLRP3, ASC, and procaspase-1, all increase in expression. The second signal is what's required to then put all of these together into a complex. The complexation of the inflammasome activates caspase-1, which is required to cleave cytokines, pro-inflammatory cytokines like IL-1 beta and IL-18 into their active forms. Once activated, they can then leave the cells to have downstream effects, not just on the cell itself, the initiating cell itself, but also on other neighboring cells. But how did we come about the inflammasome pathway? Well, we really didn't start with it. My PhD was supposed to be targeting connexin 43 hemichannels and to see whether this can prevent a disease known as diabetic retinopathy, which is a chronic disease that affects the back of your eye. And what we Just to give you an introduction on the connexin channel, first of all. So it's made of these monomers known as connexins. 6 of them usually aggregate together to form a hexamer, and this hexamer is known as a connexon or hemichannel. And then 2 neighboring cells come together and dock their connexons together to form a gap junction. And normally, these gap junctions allow cells to communicate with each other, so to pass signaling molecules, ions, and those type of things. And this is in a physiological normal setting. When you have diseases, these hemichannels sometimes don't dock together and they just open to the extracellular space, allowing unregulated flow of these molecules between the intracellular and the extracellular spaces. When this happens, then we can activate downstream signaling pathways that suggest that tells the cell that there is a pathological process happening. So what we wanted to know is, if we block these hemichannels, are we able to prevent the signs of diabetic retinopathy in both in vitro and in vivo models? And here I'll just give you an example of some of the findings that we had. Okay, so the first thing we did was we took a mouse model, a non-obese diabetic mouse, and we intravitreally introduced pro-inflammatory cytokines to the retina or to the eye. And we did this because our preliminary studies suggested to us that we needed a combination of hyperglycemia, so high glucose, and inflammation to adequately model diabetic retinopathy in these animals. And what we found was that there was an increase in inflammation. So what we're looking at here is an immunohistochemistry picture of the different retinal layers. The cell layers are labeled in blue with DAPI, and red is a marker of inflammation known as GFAP. And you can see that with no treatment, there's an increase in the red labeling, so an increase in inflammation within the retina, and that when we block connexin 43 hemichannels with peptide 5, we can actually inhibit this. And so this was told us that our drug was able to, or peptide 5 was able to prevent inflammation induced in this model compared to controls. Then we used a technology known as the Micron 4 imaging system to look at the retina in vivo in real time. And what we looked for were signs of retinal damage, such as the development of hyperreflective foci, indicated here in red, and also whether there was fluid buildup in the retinal layers, as shown here in yellow. And these are 2 pathologies that are also observed clinically in patients with diabetic retinopathy. What we found is that, again, peptide 5 was able to block the severity of this hyperreflective foci and also reduce the severity of fluid buildup at the bottom of the retina, showing that we were affecting the signs, clinical signs of DR in this model. But what we didn't really expect, and we found, is that there was an increase in inflammasome activation in this model, shown by the expression of NLRP3 and ASC, 2 of the key components of the inflammasome pathway. And blocking with peptide 5 was able to inhibit this increase in inflammasome activation. And so we asked the question, what is the link between connexin 43 hemichannels and NLRP3 inflammasome? Why is it that blocking these channels was affecting the inflammasome pathway? For this, we went to cell culture models, and we looked at ATP release. Now, if you remember, I talked earlier about how ATP is one of the activation signals for the inflammasome pathway. So it made sense to start from there. And we found that with injury, high glucose and inflammatory cytokines again, you actually see doubling of ATP on the extracellular space in the cell media. When we inhibit with peptide 5, we're actually able to inhibit the ATP release that we observed. But what's interesting is that A scrambled peptide was not able to be as effective, which means that the effect is specific to blocking connexin 43 hemichannels. When we look at NLRP3 spots on these cells, so activated NLRP3, we found that our injury increased NLRP3 and peptide 5 was able to inhibit it. So this tells us that by inhibiting ATP release, we were inhibiting inflammasome activation. And interestingly, when you put back in exogenous ATP, you're actually able to reverse some of the effect of peptide 5, suggesting that by blocking connexin 43 hemichannel, we're blocking ATP release, which is why putting ATP back in boycotts this connexin 43 pathway. And looking at downstream inflammatory markers like IL-6, the same trend holds true in that with injury, you see increase in IL-6, peptide 5 can block IL-6 release, and putting ATP back in can reverse the protection conferred by peptide 5. So we were able to build the inflammasome story into what we already know about connexin 43 hemichannels. Already we knew that connexin 43 hemichannel opening can cause an ionic imbalance, leading to the cells being unable to osmoregulate and leading to cell death. But what we have here is an addition to this pathway where we can now put in the inflammasome pathway as another way the connexin 43 hemichannel opening contributes to cell death. So that opening of connexin 43 hemichannel leads to ATP release, leads to inflammasome activation, downstream inflammation, and release of cytokines contributing to cell death. And existing therapies, especially for diabetic retinopathy, usually target this inflammatory arm, things like corticosteroids and anti-VEGF drugs. But what we're able to do is build the hemichannel blockers in as an upstream target that targets before even in the activation of the inflammation activation occurs in the cell. And when you look at the picture of NLRP3 inflammasome targeting drugs as a whole, and I'm not going to go through all of them, but we've been able to put in connexin 43 hemichannel blockers as one of the upstream targets seen here in pink for NLRP3 inflammasome and for treatment of diseases involving NLRP3 inflammasome activation. So what are we doing next? Well, here's where I introduced tonabacet, which is another connexin 43 hemichannel blocker. But the advantage of tonabacet is that it's orally available and can cross the blood-brain barrier and the blood-retinal barrier readily. It's a benzopyrene derivative, and it was actually first developed by GSK, and they completed phase 1 clinical trials for it. Unfortunately, at the time, they didn't know the mechanism of action, therefore used the wrong dose for their phase 2 clinical trials, but we've since adjusted the way the drug works now that we figured out how it works, and it's going to be going to phase 2 clinical trials in 2022 for diabetic macular edema and geographical atrophy. But in order to test tonabacet, what we did was, first of all, we wanted to use a model that was clinically relevant. So we've done a lot of mouse work, a lot of cell work, but could we develop a retinal explant model, so literally human retina, cultured in diabetic retinopathy-like conditions in which we can test the effectiveness of our drugs? So we took a human eye, opened it up, and collected explants of the retina sitting on top of the choroid and the sclera, so pretty much with all the structural integrity that normally sits in, and then cultured it in media containing high glucose and cytokines, and then tested whether tonabacet was effective at reducing inflammation within the explant on its own, or the release of inflammatory cytokines into the media. What we found was in our retinal explant, we saw an increase in inflammation, again shown by GFAP, and a lot of connexin 43 expression. And tonabacet treatment was able to bring both of them down, showing that we were reducing inflammation in this explant. And when we look at the expression of the pro-inflammatory cytokines, IL-1 beta and IL-18, both of which are directly regulated by the inflammasome, our treatment was able to reduce the levels relative to the injury alone, back towards basal levels. The same for VEGF, which is one of the key markers of diabetic retinopathy and the target for most of the drugs. So, having done all of this, what's our current research? What are the things we're working on at the moment? Well, one of the questions we have is, when is the best time to treat patients with diabetic retinopathy? Now, like I mentioned earlier, current treatments target late signs of the disease, pretty much when everything's already on fire. So the question is, can we put in an alarm system, something that allows us to know when things are about to go bad, and then treat at this point to prevent everything going on fire? And to do this, we're using human donor tissues to understand when inflammation activation occurs so that we can determine when to give our drugs. We're also looking at serum-based inflammasome biomarkers for diabetic retinopathy based on this whole idea that inflammasome activation occurs and whether it can be detected in the serum. And this is work being done by my PhD student, Charisse, and who's a second-year PhD at the moment. But we're not limiting ourselves to diabetic retinopathy alone. We're also looking at other ocular diseases, such as autoimmune uveitis, and trying to determine whether inflammasome activation occurs in autoimmune uveitis and whether we can target it either orally or with an eye drop containing tonabacet. And this is being done by Avik Shom in our lab at the moment. Finally, we're not limiting ourselves to the eye as well. We're trying to look at beyond the eye and look at whether there is a link between age-related ocular and cognitive decline and whether the inflammasome might be the pathway linking both. And we already know that inflammasome pathway mediates inflammagen, or age-related chronic inflammation, in both the eye and the brain. And they share similar pathologies like epithelial mesenchymal transition, cellular senescence, telomeric attrition, to name a few. So could the inflammasome be the linking pathway that brings all of this together? And already our preliminary data suggests to us that this might be the case, because one of the cytokines that we see, TGF-beta2, that is increased with injury and decreased with tonabacet treatment, is actually a key cytokine that regulates epithelial mesenchymal transition and plays a role in cellular senescence. So already we're starting to see signs that maybe the inflammasome could play a role in age-related cognitive and ocular decline. And I've got two master's students, Faria and Marvelous, who are working on this at the moment. In conclusion, targeting the inflammasome, we think, could have both prophylactic and therapeutic implication in various inflammation-associated retinal conditions, and now we're even moving towards age-related cognitive conditions as well. So finally, I'd like to acknowledge the Buchanan Ocular Therapeutics Unit, especially Associate Professor Ilva Rippenthal, who's the director of the unit, all of our students and technicians over the years, my mentor, Professor Colin Green, and all of my collaborators, and finally, and most importantly, the New Zealand National Eye Bank, both the staff and the donors for their support. Thank you. Any questions?
Awesome. Thanks, Lola. That was a fantastic presentation. And yeah, I never get tired of seeing the immunohistochemistry images and the OCT images. Fantastic. Yeah, we do have a little bit of time for a couple questions. So the way in which we can do this is, if you look at the bottom right, there's a little reaction smiley face. So if you could raise your hand and it pops up here, Lola can select people from the audience to ask a question. So let's do that. Let me know if there's a hand that I can't see.
Yeah, I can't see any hands.
Okay, no worries. Well, I'll just ask one question. So I might have missed this, Lola. So have you been able to isolate the inflammasome complex from any clinical samples, whether it be serum or in the vitreous or anywhere else?
Not directly. We've been looking at the cytokines that the inflammasome regulates, so IL-1 beta and IL-18 mainly. And we have found that in the vitreous of patients with diabetic retinopathy, it goes up, or they go up, both of them. And yeah, so that's mainly the direction. We're not sure. I mean, there's been some work that suggests that the inflammasome might be secreted, but there's not been a whopping lot of other studies to support that. For the most part, it looks like secreted stuff are the cytokines and the inflammasome stay put in the cells. Yeah, that's what we think anyway.
Yeah, sure, that makes sense. Okay, so if anyone else has any other questions to ask Lola, if you're a little bit shy at the moment, feel free to pop in a question into the chat. And with that, I'll say thanks, Lola, for a fantastic presentation. And I will now hand off to my colleague,
Fraser,
Elena.
I think
Sorry.
Russell's got his hand up. So.
Oh, sorry, Russell. Go for it.
Oh, wonderful talk, Lola. We've been approached recently, and there's a helicopter flying over, trying to figure out whether I'm social distancing or not. Anyway, we've been approached recently by somebody who has RP, or RP1 actually, and I thought I'd just take this opportunity to find out whether you or your group are interested in RP at all. So it's the inherited form, or one of the inherited forms.
Yeah, it's interesting you should say that, because I am a board member for Retina New Zealand, and we do a lot of work with RP in terms of that charity, and we've been in communication about what can, how to bring my research and that together in one space. We'll be interested in the long term. The difficulty with RP is because it's a genetic condition, how the inflammasome might feed in is quite difficult. And I've not done too much on the genetic yet, but I'm kind of starting on that whole telomere attrition direction at the moment. So I'm hoping that through there, I will build my, sort of genetic expertise and be able to help. But yeah, no, we've certainly been communicating about it.
Oh, okay. Just that wonderful set of methodologies and skills that you have. And, the genetics pretty simple, really, off to one side. So I'll contact you afterwards.
Yeah,
because
that'd be fun.
Yeah, they're doing something cool.
Yeah. No, that's brilliant. Thank you, Russell.
Thanks, Russell. Yeah, it sounds like a collaboration in the making, which is awesome. Sweet. So I'll pass on to Elena. You can take over now.
Thanks, Fraser. Heard it here first with the collaboration. So happy to introduce Professor Russell Snell, who you've all just met. But if you don't know him, he is based at School of Biological Sciences at University of Auckland. And Russell has the amazing ability to think of novel, creative ways to do things, which is always interesting. And I used to work with Russell and Klaus Leonard at Zealectia Biosciences, which was a subsidiary of Fonterra. So we looked at bovine genomics and identified naturally occurring genetic variations that are responsible for a whole lot of different milk traits. It was always great to go in thinking you had an idea for an experiment and then have a discussion with Russell and go, okay, well, we're not doing that, we're doing this. And just amazing to kind of plan out the whole thing and cover all bases and think of new things and new ways to do things. Today, he's always up to some cool, interesting things. There's still some bovine, a bit of ovine, and some goat, caprine, is it? And in the neurogenetics group, they're unraveling the molecular mechanisms of simple and complex neurodegenerative disorders, in particular Huntington's, Alzheimer's, and neurodevelopmental disorders such as autism. But I will let Russell tell you more about what him and his team have been up to recently, and as your talk title says, and how you're transforming lives. No pressure, Russ.
Oh, thanks, Elena. Now I need to share my screen. Blimey, you would have thought after a year or 2 years of Zooming, and as you probably all will recognise, Elena was a delight to work with, and it's really a great privilege to be able to give this talk. And this is going to be a little bit different. Why is it going to be a bit different? Hang on. How about that? I need to, somebody's saying I need to grant file access. Oh, yeah. Can you see that? Is that the right screen to be showing you? Because I did a lecture earlier this year, and as it turned out, it was the wrong screen, and I had to record the whole damn thing again, which actually I have to say was much better the second time. So this isn't going to be a talk with lots of detail. Really, it's going to be a talk around philosophy, because we're at that time of year. We've survived, and I really wanted to give you some insight in under the hood about how we think about the world and how we approach our questions. So it's really philosophical largely, and the wonderful Microsoft gave me this fantastic slide background, and it just suggested it. So I obviously knew I was science and geeky, and there it is. It's wonderful. And I'm going to suggest some areas where young scientists might go into towards the end of my talk, and with the caveat that older scientists are always good at predicting the past, not necessarily predicting the future, like all economists and some epidemiologists, and boy oh boy, we've had a dose of that over the last while. So I attempted to make this talk one slide, but it has loads of things into it. So I ended up having to make a bunch of slides, and actually I should set an alarm to try and figure out how long I'm going to go, because I do tend to witter on. Hang on, start, there we are. Okay, so where do we begin? Where do we begin in our group? So all of our work in our group has a line of sight to an outcome. We don't embark on anything for just for the sake of it, because life is short and there's loads of cool things to do that has a line of sight outcome. We're just often not sure how we're going to get there at the beginning. But I'll give you an indication of the one axiom that we stick to in our wider research group. I should say that I'm representing a whole load of collaborators, wonderful people that I work with, in particular Jesse Jacobson and Klaus Lennart, Matt Littlejohn at LIC, Alex Treverton, who's a new postdoc in our group, and wonderful collaborators all over the world. One of the joys of being in science is having collaborators, and it means you can do lots of cool things all simultaneously with lots of cool people all over the world. And I'll probably give you a bit of a flavour of that as I go. So how do we, when we start off, how can we use tricks to determine causation and predict outcomes in biology? And really that's what we're setting out to do. Although we're hoping to do a bit better than that, and fix people up if they have conditions. But mostly we've been using tricks to determine causation and predict outcomes. And our big challenge really is that often we sit. Hang on, I've got my mouse. Can you see my mouse? Oh no, you can't. There. Can you see my mouse there? Okay. Yep, we can see. Often we sit at the outside ripples, and we're trying to determine what caused those ripples. And it's a real big philosophical argument, as can you determine the cause? And in this case, it was a drip, but I would have deleted the drip. Can we determine the cause for the physiological or biochemical consequences to work back to the beginning, in particular to try and determine what the heck did what the heck to what the heck? And we spend our lives wandering around in that ripple, hoping somehow rather rationally we can determine causation. And that is a real dilemma, and very, very difficult. And largely we're too impatient to do that. So how do we separate consequence from cause? And that's as a geneticist, that's what we spend our lives doing. So particularly when actually most of the systems we're dealing with are very, very complex systems. And if we're out there measuring the magnitude or the amplitude of those waves, you know, good luck with that. And this is a sort of a semi-two-dimensional system, let alone throwing in age and biochemical background and genetic background and everything else on top of that. Yahoo! You know, one could argue, apart from basic biochemical defects, this is just about blinking impossible. Unless, of course, you're just treating symptoms, which was not very satisfying. So our problem is multifactorial, and or at least the consequences are multifactorial. So where do we start? And what we need is some form of ruler, some sort of measurer, some sort of way of determining or getting ourselves from consequence to causation. And I guess we plug for Millennium, we use loads of Millennium technologies and measure stuff up the wazoo. But you can spend your life measuring stuff up the wazoo and never get to causation. In fact, you can make careers, a whole lifetime of measuring stuff up the wazoo and never get to causation. And this is why we do what we do. How can we separate cause from consequence? And so this is what we do. This is a simplified version of what we do. And actually, mostly we've cracked all these conditions that are like this, and I'm fortunate to have been involved in a whole bunch of them and finding the genes and mutations. In this case, it's just simple autosomal dominant, but really our ruler is the genome. Our measurer is the genome. And what we do is we collect a population. In this case, it's dichotomous. You either got the disease or not, but often it isn't. I'll get on to that in a minute. And our ruler is the genome, in effect. And so we can use the genome and we can use inheritance to map locations very, very simply, whether we're using genome-wide association studies or genetic mapping. And in this case, we can do it very simply by categorizing individuals and deciding whether you've inherited the variation or not. And really, genetics over the last 30 years, maybe slightly longer, has used this methodology of inheritance, different tricks, now a whole genome sequence, to identify genes and mutations. You know, that obviously is not the end of the story. And if you find a gene and you're lucky enough that gene fits in a pathway, then it might naturally lead to some sort of therapy. But in 1993, which is quite a while ago now, we found the Huntington's gene. And, you know, I can't argue that we don't know what it does. We just know that it does a whole load of things. What we don't know is how it causes the disease. We still don't know how it causes the disease because this darn gene has so many functions. So knowing the gene itself doesn't necessarily take you to the exact causation, or at least a functional causation. But having said that, technology's moved on and loads of genomes have been sequenced. I'll talk a bit about that at the end. And we use this type of approach to identify genes and mutations in autism, and that's largely led by Jesse Jacobson. And so we can sequence an individual and we compare the sequence of that individual to databases that exist, like Nomad, for instance, and identify a variation. So without any prior genetic information, although a family is really useful, and identify with high likelihood in some cases a mutation. So the technology transformation from having a few markers to SNPs, to GWAS studies, to whole genome sequencing has been tremendous, but it hasn't got to the end of it. We're not at the end of it yet. So where do we go from here? So where we go from there is, sorry, I'll flick them all on, is I had fun doing this. Although the trouble is when you do all these animations, you can't go back and edit anything, even if you spell something incorrectly. So anyway, genome-wide association studies is the next step. And you would have seen a lot of this going on, but perhaps the implications for it are not overly obvious, and particularly in humans. In humans, the major use in my world for genome-wide association studies, and particularly as they relate to modifiers of disease, not necessarily the primary cause of disease, but modifiers of the disease, is that it can take you pretty quickly to genes and variants in those genes that form the pathway of the disease process, not necessarily the initiator of the disease process. And tremendous work has been done in this area in Huntington's disease as an exemplar of the process, identifying genes involved in somatic repeat instability. And that has identified a number of drug targets that might actually have an effect in Huntington's disease. We'll see. By implication, somatic mosaicism in the brain of that expanded repeat that causes the disease is thought to contribute considerably to the disease process. And some people think actually expansion is required to cause the disease. Not so sure about that. But what we've been doing as a group, apart from human genome-wide association studies to some extent, is we've been doing this in cows and in goats in order to identify variations that are useful to the bovine industry and to the dairy goat industry to find genes and markers that we can use for selection for these. And we've done a whole load of this. We're reasonably good at it. I shouldn't really say that, but anyway, we are. And these genes and mutations have been used for selection across these industries. So what's next? Well, this is a genome-wide association study, which can take you to a gene and a mutation. This is just an indicator of statistical significance up the side, and here's the genome lying along the bottom. And if you've done whole genome sequencing like we have in goats and we have in bovine, you can get directly to the mutation using these statistical approaches. It's really just simple statistics. But, sorry, I'm just going to make sure that I'm not watering on too long. Where do we go now? So what has it given us so far? Oh, that's lovely purple background, isn't it? We've got linked markers for selection. We've even got some genes and mutations for selection for animals. Our targets now are largely around the environment. So increasing production by decreasing environmental footprint, essentially. And in humans, kind of had a moment a few years ago that, yeah, we can discover genes and mutations and they're used for pre-implantation testing or in some families with neurodevelopmental conditions, it explains the condition and can put the family at rest. That was nothing they did, of course. It was just bad luck. It can reveal perhaps a little about the disease process, if we're lucky, the gene and the pathway. And it gives us the opportunity to discover other variants, as I've already talked about, in drug targets. But from that point on, as a geneticist, you go, oh, blinkety heck, I'm now a bystander. How can we contribute more? We don't want to be bystanders. You know, we want to be there at the end. We want to be there at the treatment part. So where we moved on to is we've been making sheep models of human disease. Why sheep? Well, any number of a myriad of reasons. Cheap, easy to breed and all that sort of carry on, but very similar brain to ours and a longer lifespan, physiologically more relevant, even though the ruminants to us. So we put a lot of effort into these animals, and we've made a Huntington's disease model, and we've used that to discover perturbation in the urea pathway and maybe reveal a treatment for Huntington's disease. But more recently, And very recently, we've got founders for Fragile X now, or a founder for Fragile X, and also founders for an early onset AD model. So that's great, and we'll form collaborations around those. But what do we do next? What can we do next as molecular biologists? So what's the world doing, and how can we join in? Are we just doing science stuff for the sake of it because we can do science stuff? You know, we're quite good at the science lark, and we get publications that sort of carry on. But we've got models that can follow the disease progression, but most importantly, we've got models where we can test therapies, and that really is our driver. And a while ago, we had this road to Damascus experience that, guess what? We could do somatic CRISPR-Cas9 therapy on somatic tissues and maybe cure a disease. Now, I would really recommend that you read this New England Journal of Medicine paper. It's really easy to read, and just came out relatively recently. And one of our own, Ed Gain, is on there. And this is a therapy for this terrible disease, transthyretin amyloidosis. Some of the clinical trials were done in New Zealand where they knocked out this gene, transthyretin, in the liver, and in effect, as far as we can tell, cured the disease. And this was just using CRISPR-Cas9. Pretty simple CRISPR-Cas9 technology. It's an outstanding paper. I really recommend that you look at it. So that kind of, I've been really reluctant to apply CRISPR-Cas9 technology other than ex vivo, where you take cells out and have a bit of a go at it, but to human disease, because having been brought up in the old school of genetics, you don't muck with somebody's genome. You certainly don't muck with somebody's germline. But goodness gracious, if you can do this sort of thing and you can cure a disease in people where there's no other cure, well, why not? So that was the road to Damascus experience relatively recently. And we're going to apply that to some of our diseases. So where is the future? Well, this is the bit that you always get wrong. So anyway, we're having a bash at it. But machine learning is the future. Absolutely no doubt about it if you've got a large enough data set. And you can study those ripples in that puddle. And you can work out to some extent, if you look at enough ripples, you will get back to some sort of causation, particularly if you throw in loads of data into machine learning. And where we can see that in these tools that are emerging, we're seeing on this Splice AI, which is incredibly accurate at picking splice sites, RNA splice sites. AlphaFold has blown out of the water a whole lot of crystallographies, because essentially you can do it mostly on the web for free in about 3 milliseconds. And more recently, just published, is this piece of software that can predict the folding of protein complexes. So you go, ooh, blinking heck, the world's changing really quickly. So okay, if we go back to, we want to be useful and we want to develop drug targets, what would I do now if I was a younger person emerging in science? Oh my goodness, you can't go past the UK Biobank as a data set. My goodness, 500,000 people measured up the wazoo for who knows what. That's going to expand with NHS data. They've just released a sequence of 200,000 people into the semi-public domain. That's genome sequence that constitute the UK Biobank, the most spectacular data set ever, and almost infinite number of possibilities to dig around in there and find interesting genetics things and things for interesting applications. So what I would do, you never know, you may even do it, is I would hook up with these people somehow. I'd buy a subscription. I'd go and hang out at the Sanger Centre or whatever it is, because there is nothing comparable in human biology to this that will get you to that initial splash in that circle that will take you directly to the gene and the mutation that will explain your area of biology and hopefully reveal but more of the mystery of human biology. But even better than that, it'll take us to therapies, essentially, which is where we want to go. There we are. Sorry, it was a bit different, but I thought it's the end of the year. We could do something different at the end of the year. And I don't have a final slide. That is my final slide. Yahoo.
Thanks, Russell. No, I love different. You know that. Yeah. Yeah. Oh, so interesting. Gets everything. Yeah. Hopefully it's got everyone's minds going. And yeah, 200,000 genome sequences. That's, yeah.
And they're going to sequence them all, the 500,000 or so, and they're going to add to them, and a whole bunch of them have MRI scans, a whole bunch of them have CT scans, there's blood samples taken up the wazoo, all their clinical records are connected, you name it. There's more American researchers are using that data set now than the UK, because there just aren't that, you run out of researchers in the end. But you can go into that, you can ask for access, you can go into it with your particular question, and as long as somebody else isn't doing your particular thing, but there's a myriad of things that one can do, then you can join in. It's just the most amazing thing. Yeah, anyway.
Yeah. We'll put it out there if anyone wants to do the hand up thing, or just unmute yourself and ask. Any questions out there?
Well, stop sharing. I'll stop sharing. There we go.
Okay.
There we go.
Or people being shy?
I don't have a question at all, Russell, but I just want to say thank you for a great talk. I'd love to get a copy of that paper on the CRISPR knockout. That's a bit of a flavor for me. It's a passion that I'm in the middle of. So I'd love to see that. And I'm so glad you've gone down the road to Damascus. I think it should be a great journey.
Yeah, I was very reluctant because of worrying about things off target effects and misuse of technology and all that sort of carry on. But actually, if the patient is in front of you and there is no other option, let's say, then I know it sounds a bit desperate, and hopefully we'll be able to do better than that. But these technologies are marvelous, and we can do cool things. So I heard this futurist talk from Israel about 5 years ago on the BBC, and I wish I'd taken his name down. I've tried searching it, but he said we're the last unmodified generation, and at that point, I thought it was such an outrageous thing to say. How dare he say that? Because it was awful. But maybe he was right. Maybe he was right.
Interesting. Have you looked into any of the the delivery mechanisms for CRISPR-Cas9? I know if you a couple of groups are talking about like the lipid nanoparticles, a bit like the way we're delivering the Pfizer and Moderna vaccines for COVID at the moment, with the thought process that having those being more targeted to some cells, or using extracellular vesicles that are targeted to particular cells to help reduce off-target effects. Have you looked into any of that?
We've given up delivering anything with a vector, so we use RNP complexes, and in some cases, so this is just for our in-lab mucking about, really, but in some cases we're getting like 85% deletions in cell culture, just utterly unbelievable. So that's in cell culture. We're using RNP complexes to make our gene-edited animals, and the efficiency is, and that's HDR in general, that is around 5 to 7%, which is fine for I think that's why we're going to focus primarily on initially dominant gain of function conditions, where gain of dysfunction conditions, cheating, where hemizygosity is not a problem. So if we can target, knock out an individual allele, which is not what they did in the transthyretin situation, they knocked out both alleles, but then they supplement them. I can't remember with what, vitamin A, I think. So it didn't matter for them. But if we can find conditions, which there's loads out there, that are dominant gain of dysfunction, that's where we'll start our mucking about, really. Yeah. Thank
you. Good luck.
Oh, well.
We've got Sylvie's asked a question, and then I'll go to you, Lola. Sylvie's asked, hi Russell, could you tell us how UK people volunteered to participate?
You know, it's a very interesting thing, but these were people all 50 and older, and I think it's a generational thing. People are really, really keen to donate or volunteer. They actually were extremely well structured. So there were loads of ethics committees prior to this. The UK does this really, really well, where there was consultation with the community and ethics, and it took years and years before any samples were collected. So the ethics are well developed, actually. We could probably use a lot of the ethics stuff that they've already developed in New Zealand, but we tend to want to reinvent it here. Did I say that out loud? But you know, they're human like everybody else is, and we can capitalize on their discoveries for application in New Zealand. The Americans are frightened, absolutely terrified of this project, because it's blowing everything they do or can't do out of the water, and that's largely because the NHS, actually, that which could have been Obamacare, but it isn't. That consolidation of information and being able to share across institutions or across hospitals or health services, that information, it's amazing. So yeah, it's ethics up the wazoo, that study.
Favorite word of the day, wazoo.
Yeah, sorry.
And I'll pass to you, Lola.
Yeah, no, thanks for that, Russell. It's actually, my question's actually linked to what Sylvie's talking about, and that is whether we can do something like that in New Zealand, so that we capture our own unique, makeup. And also, I mean, I know that already for genetic testing, you usually require seeing genetic counselors and all of that, and how that would look for, to get that kind of information in New Zealand.
So I'll start with the second part first. In our autism study for Minds for Minds, we have a strict, hard line down the middle of our study that everything we do in the lab that's in our research lab is research. But our consent process says that if people sign up for the study, they can choose whether they would like to have results. And the way those results go back is that we have this wonderful clinical geneticist involved in our study, and we give her the results out of our research study. She approaches the family and says, or we might have a finding. The sample gets taken again and tested properly through the clinical genetics process. So no research results go, apart from aggregated in papers and that sort of thing, but no research results go back, because we can't fulfill all the accreditation criteria. And also research has a different brain than a diagnostician. Juliet Taylor, sorry, is our wonderful clinical geneticist. So we separate that absolutely, and that line should never be crossed, I don't think. Never should research results go back to participants. with regard to the whole of a large study like this in New Zealand, we have contemplated that over time. We haven't made any traction, partly I think because of the expense of it. It's not millions, not even tens of millions, it's probably hundreds of millions, really, to do it properly and to have that coordinated cohort. We had imagined that because of our diverse population in New Zealand, would be a wonderful place to do that sort of thing. But whilst individuals are keen to contribute their DNA and their clinical information to that, whoops, sorry, that was, I should have switched my scope off. It's very difficult to make your way through the ethics process to get that combined group. So essentially we've given up, really. But that's all right, because loads of stuff is going on elsewhere.
Yeah.